Hello, everyone, and welcome to today's presentation, Precise Gene Editing of Human Pluripotent Stem Cells, presented by Dr. Krishanu Saha, Assistant Professor in the Department of Biomedical Engineering at the University of Wisconsin-Madison. I'm Marian Pekarchik of Thermo Fisher Scientific, and I will be your moderator for today's presentation. Before we begin, I'd like to remind everyone that this event is interactive. We do encourage you to participate by submitting any questions you may have at any time during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your question into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know of any technical difficulties. I would like to introduce and give you some background on Dr. Saha. Dr. Saha is an assistant professor in the Department of Biomedical Engineering at the University of Wisconsin-Madison. He is a member of the Wisconsin Institutes for Discovery, the Carbone Cancer Center, and the Stem Cell Regenerative Medicine Centers, as well as the National Academies Forum on Regenerative Medicine. Prior to his arrival in Madison, Dr. Saha studied chemical engineering at Cornell University and at the University of California in Berkeley. He was a Society and Science member of the Branco-Weiss Fellow at the Whitehead Institute for Biomedical Research at MIT and in the Science and Technology Studies program at Harvard University. His major goals of his lab are to involve gene editing and gene engineering of human cells found in the retina, central nervous system, and blood. I will now turn the presentation over to Dr. Saha.
Thank you. That was a very nice introduction. Today I'll be talking about engineering human pluripotent stem cells. A lot of this work is coming out of projects supported by the National Science Foundation as well as the National Institutes for Health. In particular, there's a consortium, Somatic Cell Genome Editing Consortium, and Cell Manufacturing Technologies Center that we're part of for several projects. And on this image, you'll see an image of the Cas9 protein together with an RNA, as well as a DNA template that's a useful formulation that we use, and I'll be describing that strategy as well as another strategy that seems to work quite well in our hands for human pluripotent stem cells. So I want to first give a motivation here. This community, and I assume the audience, is very familiar with human stem cells, but in particular we're focused on human pluripotent stem cells that could be derived from human embryos or through reprogramming to make induced pluripotent stem cells. And these can be patient-specific, so some of the applications here have been to model diseases, use them for drug discovery, use them for toxicology at the stem cell state or after expansion and differentiation, as shown in this cartoon. What we've been interested in is using this type of platform for testing genome editing strategies. There could be a number of reasons why you want to do this. Perhaps you want to change a pathogenic variant that you've seen in your pluripotent stem cells to understand its function. Or to provide an interesting isogenic control for some of these assays. Or they could be actually useful as cell therapies or resources for tissue engineering that actually get transplanted into patients. Regardless, I think one of the ways in which our lab has been working with these resources is to think about how we could make better genome editors using these human cellular platforms. So there's a number of different ways, and I've outlined 4 here on this slide. First, I think it provides a benchmark, and I mentioned correction or knocking in or writing in new types of genetic variants that you want to see within your cell lines. You can also compare the effect of gene augmentation side by side. That could be through viral vectors or other strategies. it's also a relevant human system to think about adverse events from applying high doses of genome editors. You can also test in a patient-specific manner here in 3 if there are particular single nucleotide polymorphisms or SNPs that could lead to new recognition sites for genome editors. And lastly here, you can think about and explore the effects of host factors that could vary from cell type to cell type because you are working with a stem cell platform that has the same genetic sequence that you can differentiate to either a progenitor stage or a mature cell type stage and see whether it matters and how it matters to do genome editing at those different cell types. Notably because the trafficking machinery, the receptors on these various cell types could vary, as well as DNA repair machinery that's relevant for many of the nuclease-based genome editors. So I want to move here to the system that we work with most often, which is the CRISPR-Cas9 system. You see it's an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. And Cas9 is right now the most common protein, which is a CRISPR-associated protein that allows us to specifically cut the genome. And this system is shown in cartoon form here on the slide, taken from the reference at the bottom. And it's really coming from a bacterial immune system that's used to degrade invading DNA. I'm not going to go through really the details of the system since the review is there. There's actually a very good Radiolab link that's on the slide that describes the very interesting story of how the system was discovered and developed. But regardless to say, there's small, short, hairpin-like RNAs that's shown in part B on the top right that gets recognized by Cas CRISPR-associated proteins that ends up cutting foreign DNA, and foreign DNA in part C is shown by the invading phage. So this system has been engineered to work in human cells, and it can cut specifically based on the RNA sequence. And one cartoon here that illustrates how the CRISPR system works. The blue protein here is Cas9. It's loaded with a RNA that's recognizing the gray genomic DNA sequence. And what the Cas9 does is it makes a DNA double-strand break. And this break can be repaired by the cellular machinery primarily through 2 DNA repair pathways. One is non-homologous end joining, which is labeled on the left here as error-prone end joining. And what that results in is small, on the order of a few base pair, insertions of random bases or deletions of the ends of the cut site that generate a wide spectrum of sequences, specifically where the cut was made. There's also the pathway on the right called homology-directed repair, and you see a differently colored strand there that's labeled as SSODN. That's for single-stranded oligonucleotide. And if that's designed correctly, such that parts of it are homologous to the ends of the cut site, you can actually incorporate the exact sequence that's written into the exact sequence that's on the template, which is written into the genome. To give you an example of what this looks like, the wild type sequence here is on the top, and the cut that we make using the CRISPR system is between the C and the A, and you see a lot of dashed lines there. And so the template, that SSODN in this experiment, has the wild type sequence, but it has this blue knocked in sequence that we're trying to write into that specific spot. And so all of the outcomes here at the bottom, the blue as well as the orange, are outcomes that we get after we use this genome editing system in human pluripotent stem cells. And you can see that we get in some of our reads the precise insertion of what we wanted from the template, that's in blue. But we also get these other reads, and you could have the insertion correctly, but upstream there's an underlined C in orange that is a mutation at that spot. You can also have a deletion at that spot, or you can get partial incorporation, which is the second from the bottom, where about 5 bases or so have been deleted in the insertion. And then the very bottom, NHEJ for non-homologous end joining, you can have the ends chewed away from the cut site. And so if you're trying to make a cell line that has this blue sequence specifically at the cut site, all of these orange sequences are not what you wanted, and they could have important differences from what you intended. So we, in our lab, call this precise versus imprecise editing. And we recently wrote a review that describes precision in genome editing, and what I just described is highlighted as number 2 here in this cartoon among 4 different ways of thinking about precision. The most common way that people think about precision of genome editing has been to make a cut where you want it and not cut elsewhere in the genome. So these other areas elsewhere in the genome have been described as off-target sites. And a lot of the precision that you can achieve using a specific cut, it has to do with engineering the nuclease, a little bit of engineering the guide sequence in the CRISPR system. That's really not the focus of my talk today. We're much more interested in part 2. The other ideas of precision, just to describe this slide here, in 3 is that you also want the edited sequence to be expressed properly. So you can you should think also about precise transcriptional control of your edited sequence, as well as another idea when it applies to the body of editing the right cell type in the right tissue. So these are all different ways, at least in literature, you can see precise genome editing being described. Today's talk and what I'll focus on is really number 2, to be able to do the precise sequence level knock-ins or changes where you're cutting. So 2 methods that we use routinely now in the lab are a transient puromycin selection method that I'll talk about in the first half of my talk, and then secondly a RNA simplex method, and this takes advantage of an RNA aptamer that brings the donor template together with the cutting machinery. So this first puromycin selection method we've been using mostly on pluripotent stem cells that have come from patients with a number of disorders, and we focused on a specific gene called BEST1 in a retinopathy. This in the light green circle on the bottom is one of the genes implicated and has been seen in patients that have vision loss. And what this gene is that it has, and if you look at the protein structure of BEST1, you can see a number of mutations in red, and a patient that's been diagnosed with this typically has a autosomal dominant mutation. And what we wanted to do in our lab, and what we have done now, is to specifically fix some of these red mutations in the stem cell lines that we get through reprogramming these patients' blood samples. After we reprogram, we also generate retinal tissues, optic cups, as well as pigmented epithelial layers. These are the kind of two tissue constructs that simulate what the retina is inside these patients. This is through a collaboration with David Gam's lab here at UW-Madison. And the very first times we tried to fix these mutations using some of the standard CRISPR tools, we had very low efficiencies, and we took a step back and we started doing some studies on human cell lines after delivery of the CRISPR system. And one study that's summarized here in the reference at the bottom is that we loaded a Cas9 GFP mRNA to encode the cutting machinery, in vitro transcribed short guide RNA, and various types of transfection materials. And we did this in kind of a medium throughput way, such that we can watch in cells that have that are receiving these packages of the genome editors. And we have this platform where we can look over time at the same cells in about small subpopulations. You can see on the right these little dots on the surface of our slide have a salt and pepper type of expression of M-cherry. And so the system here, and it's described in the paper, is that we have an M-cherry fluorescent tag that we're trying to edit, and by watching the editing of M-cherry, we can see which of these cells and when do we get genome editing. And so that's summarized here. As I mentioned on the top, we're delivering basically an all RNA form of the genome editor. And because there's a GFP fusion in this mRNA, we can see where the protein is. And because the cells have a M-cherry here in the genome, as shown in part E, whenever we get successful genome editing, and if we wait for protein turnover to happen, we see a loss of that M-cherry if the CRISPR system is working. And so what we found is that if there's quite a bit of heterogeneity, and so some of these cells get quite a bit of GFP, that's shown on the right, and that's since it's a fusion, is also tagging Cas9. And then some at the top here get less translation of GFP, and they have less editing. And that's about 4 days after delivery, we see a lot more M-cherry in these cells at the top, meaning they weren't as edited as the cells at the bottom, where you see mostly just blue cells without that many M-cherry positive cells. And so if you look at thousands of these types of spots and start to make correlations about which materials can deliver effectively and what doses of each of these components, the mRNA encoding Cas9 as well as the short guide RNA, you can start to see trends. And this is again from the same paper. We see some of these transfection reagents working quite well. And when we look in a quantitative way and try to predict which cells will be edited, based on some characteristics of expression and cell density, we found that cells that express Cas9 and translate Cas9 early have more gene editing, and we find that cells that actually do not proliferate early, so that's the delta cells on the right as one of our coefficients in this regression model, in the first 24 hours, those also have a higher editing. And our thought here is that there's probably a stress that's induced by cutting of the genome, as perhaps also by delivery. And if we're able to identify these cells that are slowing down, but also have a lots of Cas9, those are the ones that would be edited most frequently. So we applied this to human pluripotent stem cells, these insights, and we had to change a little bit of how we delivered the CRISPR system, notably because those mRNA formulations did not work that well in human pluripotent stem cells. All of that work was with human embryonic kidney cells. But regardless, I think the idea that you deliver Cas9 at high levels early after delivery, and then slow down the cell cycle, these are things that we were able to achieve using a plasmid form that encodes the CRISPR system. So on the top left here, there's a plasmid that has Cas9 expressed with a 2A sequence and then a puromycin's resistance gene packed. And so whenever Cas9 is expressed and also translated, you will have this resistance protein inside the cell. We also co-deliver this single-stranded oligonucleotide template, and we rely on electroporation or nucleofection to get all of this into human pluripotent stem cells. And importantly here, the puromycin here is not encoded on the donor, and that allows us to do only a transient selection over the first 4 days or so. So we start at day 1 and we end at day 4, 3 days of puromycin selection. And that's simply by adding puromycin to our media. And on the right here, you can see a summary of our editing results, which is as you move from zero, no puromycin in the media, you get almost all of your cells. There's a very, I think, small green bar there. Almost all of your cells are not edited. As you start increasing selection, increasing for the cells that have high levels of Cas9, you get more homology-directed repair, that's the precise editing outcome, and more indels, that's the imprecise editing outcome. And there's kind of a sweet spot where you get 4-to-1 or higher type of ratio of the precise edited clones versus the ones that have imprecise edits. And so a lot of the data is summarized in this stem cell reports paper that's listed at the bottom by a talented now graduated student, Ben Stayer. We also have been looking in some of these patient-specific disease models that I mentioned for the BEST gene, and we're able to look how specific the CRISPR system is. And because in some of these cases for a dominant allele, you have one wild-type allele and a mutant allele, we can make and design CRISPR genome editors to selectively degrade one allele versus the other. So we have cells that are from healthy individuals, where you have 2 wild-type copies. And in this case, we're delivering virus for the CRISPR system in stem cells. And this is another delivery system that works quite well if you just want to cut the genome and rely on imprecise genome editing. This slide is mostly to show that you can achieve mutant-specific allele editing, where if you make these guide RNAs specific to the mutant allele in blue, or the mutant allele in red and the wild-type allele in blue, you get editing, as shown on the y-axis, specific to that allele. And you can also, if you do next-gen sequencing, see which of these edited alleles, exactly what sequence you get, if it results in a frameshift mutation that should result in nonsense-mediated decay if it's not in the last exon of the gene. So this is kind of a pretty popular way that we've been using on stem cells for knocking in variants where we have cells that grow quite well from patients. Another alternative is to use a RNA aptamer that doesn't rely on any plasmids or mRNA. The advantage here is that there's probably a lower likelihood of random integration of your plasmid into the genome. And if that's a concern for you, you could use potentially one of these protein and short RNA-based methods. So again, to summarize, we're still looking at this challenge here of making more precise reads in the clones that you get. And our strategy here was to think about co-delivering the donor template with the cutting machinery. And we exploit this connection here that's shown in the cartoon that is called an S1M aptamer, that's specifically shown in red. This aptamer is from some studies in 2013 that found essentially a biotin mimic in a short RNA sequence. And the first version of this was called S1, shown on the left. And the blot at the bottom shows that as you increase the amount of streptavidin that would mimic the biotin streptavidin binding, you get more and more binding of the RNA aptamer to streptavidin. There's a modified version called S1M that stabilizes the stem part of this structure, and it has a higher affinity for streptavidin, and that's shown in red. So we took this sequence and we looked at the crystal structure of Cas9 and together with the guide, and found a few places where we can introduce this aptamer. We put it into a few stem loops and at the end, and we call these version 1, S1M, sgRNA 1, 2, and 3. And we wanted to see if this actually does bring together all the components that we wanted to together, and we relied on this technique called dynamic light scattering, where you can look at the correlation in intensity and determine size of particles after shining a laser in a solution of them. And this slide is a bit busy, but I want you to focus on the green trace here that's shown, and on the x-axis is shown as the radius of the particles, the average radius, and that's in nanometers, and on the y-axis is normalized intensity. And so the green is what we call the simplex, where it has the aptamer, the Cas9, the streptavidin, and whenever we take away one of the components or multiple components, we get much smaller particles. We wanted to see whether these particles stay intact inside the cell, so we can look at pluripotent stem cells after we deliver these simplexes, and we can look by staining for Cas9 protein and streptavidin, the two protein components here. On the top is a version where we take away the aptamer, and on the bottom here is a version where we have the aptamer, and there's more yellow dots and speckles inside the nucleus specifically of these cells. And this is a zoomed in version of the same image. And if we look at the correlation of the co-localization of streptavidin Cas9, whenever we have the S1M aptamer, we get higher correlation inside these cells. And this is described in this paper at the bottom, published last year. So does it matter where we put this aptamer? It does to a moderate effect in all cases after we deliver these simplexes into human pluripotent stem cells. We get an increase in the precise editing outcomes. And so you'll see a number of these types of slides that come from the paper where on the y-axis you have a ratio of the precise over the imprecise reads that we get in sequencing after we sequence the cells. In this case, it's about 6 days after electroporation. So the three different aptamer locations and the three different simplexes all seem to increase the ratio of precise to imprecise. Putting the aptamer in locations 1 and 2 in the previous slide seemed to help the best. And so all of the data from now on is by using the aptamer in position 1. And so we tested it at a number of different loci. This is at the blue fluorescent protein transgene, where we've tried to switch 3 nucleotides. And this simplex seems to help almost 18-fold. So now we have 4 times on an absolute level of precise reads to imprecise reads. And then we always compare against the standard ways of delivering everything without the aptamer. And we've looked at larger knock-ins, so 18 nucleotides, and we've taken away systematically other parts of the complex. Only whenever we have the full complex do we see the increase in precision. And we've also done it at the EMX1 locus that's been used in other genome editing studies. Here we've looked at GAA as a gene that's involved in a disorder called Pompe disease, the newborn disease, and we've kind of flipped this chart around because we were playing with different ways of modifying the single-stranded oligo, and where it could be designed to have the biotin on the 3' end or the 5' end, or it could be symmetric around the cut site or asymmetric around the cut site. All of that data is not shown here, but we did not find a really obvious effect from tethering the donor in various ways. In all cases, when we co-delivered, we had about a 10-fold increase whenever we used the simplex versus not. And so on the left portions of this graph, you'll have precision to imprecision ratios below 1, and whenever we use the simplex on the right, we get in the about 4 to 9 range of precise reads for every imprecise read. So some people wonder whether off-target events are different when we use this strategy, and we looked in a fairly limited manner at 5 off-target sites for several of the simplexes that we made. In most cases, using this Sanger sequencing method, PCR amplicon Sanger sequencing method, we don't find any obvious off-target events when we use our simplex. In one case, for one off-target, in our EMX1 strategy, the standard way of doing genome editing had some off-target events. So, I think more studies could be done, but we don't see an obvious increase, if not even decrease, in off-target events. One of the other nice tricks that we can do with our simplex is that we can bring in other cargoes, because streptavidin has 4 binding sites, and we're only using 1 for the donor. We can attach a quantum dot such that we can see where the genome editing machinery is, and we can attach it through different types of linkers. In one case, the linker did matter for how effective the genome editing was occurring. One application of this has been to make 2 different edits in the same cell, and what you can do is, by having a fluorescently tagged version of streptavidin, you can have one CRISPR machinery simplex here directed against one locus. In this case, on the left, the green is against an EMX1 locus with a particular repair template, and the red simplex, which is a red tag streptavidin, would be against a different locus. In the right here is shown blue fluorescent protein. So if you mix these up in a cuvette and then do electroporation, you can see some instances of cells that have the simplexes inside the nucleus again. If you do FACS and try to purify out cells that have both green and red simplexes, double positive, or the red simplexes only, or the green simplexes only, or no simplexes, and then do sequencing, you'll see the expected trends in editing outcomes. So the double negative have low amounts of edits on both targets. The green ones have a lot more green precise edits and no detectable red edits. The red positive fraction had a lot more red edits and lower green edits, and then the double positive had higher levels or high levels of both. A lot of this depends on how stringently you gate, but at least you can enrich for the types of edits that you're looking for. And so this is a kind of summary of, I think, the types of outcomes that we get when we use this system. And I want to kind of quickly go through in about a few minutes here how we've applied this system to a few cell lines, one in particular for Pompe disease. And I mentioned Pompe disease is this loss of this GAA inside cells, and the muscles in these typically babies are not very strong because there's a buildup of glycogen inside the lysosome. If we're able to edit them, which we have been able to do, you would restore activity of this GAA in the lysosome and see more glucose inside the cell. And so these are challenging edits to make because you have 2 different mutations on maternal and paternal copies of the genome. And so allele 1 here has a mutation, G to A mutation at this in red, and then the other allele has a mutation earlier called deletion of T. So these are compound heterozygote mutations, and so we design simplexes that repair both of these problems, and we take advantage of both of the platforms that I talked about, that high content platform that was those spots that I described about delivering mRNA, as well as the simplex, and we were able to correctly isolate edited iPS clones that had all of the markers of pluripotency, and when we did sequencing, had every possible repair here. So you could have a repair on one allele or the other, and you can have repair of both alleles. And I'm not going to go through all of these sequencing traces because of time, but it is the sequencing traces look as expected by looking at these two alleles. We also wanted to check that we're not causing a large-scale genomic deletion or insertion, and we did this by doing some long-range PCR, and that was done across both of the mutations, and in all cases we saw the bands as expected, no problems in karyotypes, and proper pluripotency markers. So one thing that you can do is see that this edited alleles are properly expressed, and it's a little challenging to show, but we've done some deep sequencing of mRNA, and we actually see in blue overrepresentation of the edited allele. So when we sequence, in this case at exon 9, so at the mutation on allele 1, we see overrepresentation of the edited allele in the mRNA. And when we look and deep sequence at the mutation location for allele 2, we see a similar trend, overrepresentation of the edited transcript. And then we can also look at the 3' junction. In that case, we can't distinguish which allele is coming from. We again see a higher level when we do correct both alleles. One of the advantages of doing this type of gene editing versus doing gene augmentation is that you're not flooding the cell with one single open reading frame, and you potentially can have native protein processing at endogenous levels, and that's exactly what we see when we do the Western blot. And in this case, for Pompe disease, we are also interested in how much excess enzyme gets secreted from cells, and we've differentiated these cells to cardiomyocytes, and we see actual cooperativity, meaning that we get a synergy when we correct both alleles. So that's, I guess, a small story, really quick story of how you can do multiplex editing to change, in this case, 2 alleles in the same cell. So to summarize here, because I want to have some time for questions, I think pluripotent stem cells can be an important preclinical platform to test genome editing strategies. Precision can be increased by transient selection or co-delivery of a genome editor. And I think some of these reagents are nice in that for the aptamer case, that they can be quality controlled and developed potentially as an off-the-shelf reagent. So I'll just end here by acknowledging the team that's really pushed this forward, notably Jared Carlson-Sturbermer, who's now at Synthago, and Ben Stayer, who is in medical school at the moment. Be happy to take any questions.
Thank you, Kris, for that great presentation. We can now begin with the live Q&A portion of the webinar. I do see that we do have a lot of questions that are already in queue, but if you do have a question you'd like to ask, please do so now by clicking on the Ask Question drop-down box located on the far left of the presentation window. Type your question into the box that appears on the screen and then click the Send button. So let's get started. I have a question here. First question is, have you reduced the off-target effects significantly with the use of the S1 Simplex?
Yeah, I showed that slide where we saw less off-target effects at one off-target site. We don't have, deep genome-wide sequencing data, so I'm hesitant to say that that's going to hold up for every particular strategy. But I do think, there are good studies about limiting the amount of Cas9 expressed and delivered inside the cell to get the most kind of hit and run at the target, and such that Cas9 and the machinery isn't around to bind to off-target sites. And that's where the Simplex strategy may be potentially a bit better than the transient selection strategy, because I think the Simplex is only around for probably the first 2 to 3 days at most, whereas I think the transient selection method is longer. Most plasmid-based systems can take a few extra days of Cas9 expression. So that's one of the reasons why my lab really likes some of the protein methods, because you don't have to rely on host machinery, but you also have a limited window in which cutting is happening, just because the protein is being degraded.
Okay, great, thank you. We have another question here, and the question is, how will epigenetic memory affect patient-derived iPSC cells compared to embryonic cells?
Well, I think that's something that we didn't directly discuss today, but in memory, if it can bias the types of lineages that you see, as well as particular genes that get turned on or turned off through the reprogramming and differentiation process. So we have not seen much epigenetic memory, at least in the types of assays that we've been looking at, but we haven't really looked very thoroughly also genome-wide for memory. Okay.
Here is actually here's a question actually that with relativity is there a possibility of undesirable insertions using the Simplex methodology?
Yes, the possibility is certainly there. It is hard to track, and this is a challenge for almost every single strategy that relies on a donor template. So you could design PCR probes and look for them through some sort of amplification step in the genome afterwards. We haven't had success in those types of assays, and therefore we don't have great data to talk about off-target insertion of these SSODNs. I think there's a general sense in the field that the single-stranded oligos insert less than the double-stranded templates, and that's another reason why we like the kind of single-stranded approach.
Okay, great. There is another question here. CRISPR has an obvious advantage over lentivirus-derived iPSCs reprogramming, but can CRISPR be used for recombining or adding transcription factors into DNA?
So, I mean, I think the there's a few recent papers that talked about using a dead Cas9, meaning one that doesn't cut the genome, but is fused to transcriptional activators or epigenetic writers. I think there's one story out of Duke, out of Charlie Gerspech's lab, that showed that you could reprogram cells using that type of dead Cas9 strategy, and there's a few other reports out there. Trying to do precise genome editing to write in a transcription factor for reprogramming, I have not seen that. There are, I think the episomal strategies that we use avoid some of those integration issues when you want to write in a transcription factor. So
right.
I would point, I think if I wanted to do reprogramming using CRISPR, I would probably go to a dead Cas9 strategy.
Okay. And I have, I think we have time probably for one or two more questions I do see here. Do you foresee therapeutic use of iPSCs in the near future beyond disease modeling roles?
Yes. There are clinical trials, I think, already underway in the eye. And there are also biotech companies and others trying to generate a kind of allogeneic approaches from iPS cells such that you can modify the immune response to have a kind of a more universal transplant cell. So I think there's a lot of interest in being able to generate interesting cell therapies from a pluripotent stem cell platform.
Okay. And yep, I have another question here. Do you have reduced the off-target effects significantly with this new Simplex methodology? You probably have already answered that, but that is a question here.
Yeah, so I think the off-target effects, going back to the kind of 4 ways of precision slide, is something that we looked at, and I think we don't have great evidence to say that it's lower. I think what is safe to say and more conservative to say is that it's comparable as standard methods. I think the major story here that I wanted to convey is that the on-target spectrum of sequence outcomes that you get is much more defined specifically to what you wanted in your repair template using the strategies that I talked about. And therefore, at least in the stem cell workflow, you have to screen through a lot less clones, and that I think is one of the bottlenecks here, that because stem cells grow relatively slowly compared to some of the other cell lines, and can also obtain genomic instabilities upon extended culture and stress, it's favorable to be able to have a more condensed, more efficient workflow to find the edits that you get at the intended target site.
Okay. Great. Thank you. So with that, I think we want to thank you, Kris, for your presentation, and let people know too that before we go, that the seminar webinar will be available for on-demand viewing through February of 2019. So we do thank you again for joining us, and we hope that you do check into other events today throughout the rest of today's event and enjoy your viewing.
Thank you, everyone.
Thank you, Kris. Yep. Thank you.